Hey, Hammy here, back with part 1 of section 7.2, in which we begin to look at the process of taking the instructions, the genetic code on DNA, and turning it into the protein out in the cytoplasm. Okay, this video is going to cover that first step of the central dogma called transcription. Okay, remember the central dogma of molecular biology states that DNA is made into RNA, specifically messenger RNA. Okay, that first step, transcription, is this part right here. So it's happening in the nucleus where we find the DNA, because remember, DNA does not leave the nucleus. It's protected in there. Okay, and it's a small piece of it, wherever that gene is for a certain protein that the cell needs, it's gonna read that and code it into a messenger RNA. Okay, the process of transcription has 3 main steps. The first step is initiation. Okay, wherever that gene for that protein is, there's going to be a region that starts early on in that DNA strand called the promoter. Okay, that promoter is going to say, hey, over here, here's the gene. This is what needs to be read. Okay, now notice it's only on one side of the DNA here. Okay, so on that side of the DNA is we're going to use that code, okay, to make a protein. All right. So at that promoter is where this enzyme called RNA polymerase, because remember enzymes end in ase. It's similar to DNA polymerase that we talked about in DNA replication, only instead of plugging in DNA nucleotides, it's going to plug in RNA nucleotides. Okay, after the RNA polymerase is attached and the DNA double helix unwinds, okay, and the hydrogen bonds in the middle break apart, step 2 is elongation. Okay, it gets longer. So as the mRNA, okay, reads from the start sequence up here, okay, the promoter region right here, as it reads along the DNA, the RNA polymerase is going to plug in, okay, the complementary RNA base code. So if on the DNA it said A, T, G, C, the mRNA, it's going to build the mRNA strand over here, RNA. It's going to, wherever there's an A, instead of putting a T, remember RNA has uracil instead of thymine. So it's going to plug in a uracil. Wherever there's a T, now there's a T because this is still the DNA, the original strand over here. It's going to match that with an A on the RNA. G goes to C, and C goes to G. So it's just going to read along the mRNA down here, and you see this strand of mRNA forming here, messenger RNA, forming as it reads along, it gets longer, hence the name elongation, right? Okay, the third and final step is called termination. Okay, at the end of the sequence, this green region right here, there's kind of a termination or stop region, okay, that tells the RNA polymerase to detach, and the mRNA transcript, okay, the messenger RNA transcript here is also going to detach from the DNA. Okay, and the reading of that actual gene for the protein. So now we've copied the recipe from the DNA, the recipe for that protein, we've copied from the DNA onto this strand of messenger RNA here. Now remember that transcription is happening in the nucleus, but before it can go out to the ribosome to make the protein, okay, it's processed. In eukaryotes, the mRNA is modified before it leaves the nucleus and goes to make that final protein. Okay, what happens is these modifications, based on how it's processed and edited, can make more than one possible protein from that one recipe. So think of it as some slight variations that can occur based on how it is edited from the DNA till it leaves the nucleus here and goes through the nuclear membrane out to the cytosol or cytoplasm and made into this final protein structure down here. Okay, one of the first types of editing that are done is called splicing. Okay, just like you splice a wire together, there are these regions in the mRNA called introns. Okay, we used to call junk DNA, and we thought it was just discarded. Okay, now we know that it's not really junk. It's just used for different purposes. Okay, so in the certain protein that we're trying to make, we will cut out certain introns, leaving exons left. I like to remember that the introns are cut out and the exons are expressed as proteins. So the actual things you need for the recipe are the exons. Okay, so if you look down here at the pre-mRNA transcript, okay, it has exons and introns, purple and kind of that salmon color. There are little small ribonuclear proteins, S, N, okay, R, small ribonuclear proteins. I just usually remember it as SNRPs, okay, SNRPs. Or some books call them ribozymes because they're little RNAs that act like enzymes. They will come along, and you see these look like little purple blue commas down here. They are going to cut out the introns. Remember, the introns are purple. So it's cutting out the intron, and then it'll splice the exons back together. So when we take out this, okay, what you end up with is the mature mRNA transcript down here where all the introns have been taken out. Okay, it's like going back through your essay and you say, oh, there's extra words in there, and you take them, or that paragraph's not really good, so you just chop, highlight it, hit delete. It takes it out, but it doesn't leave a blank space there, right? On a word processing document, when you hit delete, it closes up the space, right? It goes back. Same thing happens in the RNA here. The second type of processing that can be done is called editing. Okay, editing is when actual changes are made in the mRNA. There's a protein called ApoB that's found in the blood that helps carry lipids. Scientists have found slightly different ApoBs. Okay, and the one it's edited, and it actually puts like a termination or stop sequence in it. And it actually makes one of the ApoB proteins shorter than the other ones. Okay, so they have similar functions, similar structures, but sometimes these mRNA transcripts are edited and they make slightly different proteins based on what they might be needed for. And the final type, before that mRNA can leave the nucleus, polyadenylation. Polyadenylation. Poly means many, adenines. Okay, it adds a tail. It's kind of easier to say a poly-A tail on the end of the transcript. So down here we see exons and introns. Okay, the introns have been cut out. Okay, so they've been processed, spliced, okay, and it's been edited. So here's our 3 exons that are expressed to make that protein. Okay, here's the recipe right here. And on the front end and the back end, we add a poly-A tail, just a bunch of adenine nucleotides, A. And we often add like a 5' cap, often made up of guanines. Okay, a guanine cap sometimes you'll see. This just helps protect, this kind of code in the middle here. There are enzymes out in the cytoplasm, okay, that will kind of eat away at these RNAs. So this helps it last longer out in the cytoplasm because it's not going to erode back into where the important information is there in the recipe for the gene. Okay, so after this editing, now we are ready to take this mRNA transcript. Okay, we're ready to take it out to the cytoplasm. Okay, where it's going to be read at the ribosome and be made into a protein. Okay, so this first part is just transcription, where we go from DNA and it's transcribed into the mRNA, the first part of the central dogma. Okay, in the next video, we'll look at the second part of the central dogma and how this mRNA is then taken out here and made into that final protein called translation.